Hello, and thank you for joining me on this microscopic journey. Tonight, we will slowly drift into a state of calmness and explore the invisible world where bacteria reign supreme. These tiny organisms, often unseen by the naked eye, have an extraordinary ability to thrive in even the harshest conditions. Picture yourself moving through the ethereal depths of an underwater world, where strange, vibrant shapes pulse softly in the currents. Bacteria have been on Earth for billions of years, surviving in extreme environments, from the boiling springs to the frozen depths of the polar ice caps. Their adaptability is nothing short of magical, and tonight we'll uncover just how they do it. If you enjoy these relaxing moments, please consider leaving a like, subscribing, or sharing your thoughts below. Your support is deeply appreciated and helps others find their way to this peaceful space. You can also turn on the notification so you don't miss any of our future journeys together. But for now, just breathe. Let your body relax, release any tension, and allow your mind to drift. Let's begin. Bacteria are among the earliest life forms on Earth, evolving over 3.5 billion years ago. These microscopic organisms were crucial in shaping the planet's atmosphere, producing oxygen as a byproduct of photosynthesis long before plants evolved. Their influence on Earth's ecosystems is immeasurable, forming the foundation of many food webs and ecological processes. Despite their microscopic size, bacteria are incredibly resilient, able to survive in the most extreme environments, from boiling hot springs to deep-sea hydrothermal vents. Some bacteria have the extraordinary ability to survive in outer space. In 2007, researchers found that bacterial spores could endure exposure to the vacuum and radiation of space for several years. This remarkable survival capability is largely due to their tough outer spores, which shield them from the harsh conditions of space. This discovery hints at the possibility that life, in some form, could exist elsewhere in the universe, carried by bacteria in spacefaring meteors or comets. One of the most intriguing bacteria is Deinococcus radiodurans, often called Conan the bacterium. This microorganism is famed for its ability to withstand radiation levels that would be lethal to most forms of life. In fact, it can endure radiation doses 1,000 times higher than a human can survive. D. Radiodurans is also known for its ability to repair its DNA, even when it has been shattered into pieces by radiation. Bacteria can adopt a variety of shapes and sizes, from the typical round cocci to rod-shaped bacilli and spiral-shaped spirilla. These shapes are more than just cosmetic. They play important roles in the bacteria's ability to survive and thrive in their environments. For instance, the spiral shape of Helicobacter pylori, a bacterium found in the human stomach, helps it navigate the thick mucus lining of the stomach and burrow into the stomach lining where it can cause ulcers. The world of bacteria is not limited to Earth's surface. Some bacteria live deep within the Earth's crust, miles below the surface, in a habitat called the deep biosphere. These bacteria consume minerals rather than sunlight or organic matter, and their existence has opened up new possibilities for life in extreme environments, including other planets or moons in our solar system. One of the oldest forms of bacteria is Cyanobacteria, also known as blue-green algae. These bacteria played a pivotal role in the Earth's early history, responsible for the Great Oxygenation event about 2.4 billion years ago, which resulted in the increase of oxygen in the Earth's atmosphere, making life as we know it possible. Today, Cyanobacteria continue to thrive in oceans, fresh water, and even deserts, where they play crucial roles in nitrogen fixation and oxygen production. In the soil, bacteria help break down organic matter, recycling nutrients like nitrogen, phosphorus, and carbon back into the ecosystem. They work alongside fungi and plants in an intricate dance of decomposition and nutrient cycling. This process is vital for maintaining soil health and supporting plant life, which, in turn, sustains animal populations. One of the most astonishing abilities of bacteria is their ability to communicate with each other through a process known as quorum sensing. Using chemicals called autoinducers, bacteria can detect the density of their population and coordinate behaviors such as bioluminescence, virulence factor production, and even the formation of biofilms. This synchronized behavior is crucial for bacteria to thrive in colonies and adapt to environmental changes. Bacteria are known for their ability to share genes, including those for antibiotic resistance, through horizontal gene transfer. This allows bacteria to quickly adapt to new challenges, such as the presence of antibiotics, by exchanging genetic material with other bacteria. The rapid spread of antibiotic resistance is a major concern in modern medicine, highlighting the importance of understanding bacterial genetics and how they evolve. In the human body, bacteria outnumber human cells by a factor of 10, forming what is known as the microbiome. This microbial community plays a crucial role in digestion, immunity, and even mental health. The balance of bacteria in the gut can affect everything from mood to susceptibility to disease, which is why maintaining a healthy microbiome is vital for overall well-being. Bacteria also play a role in the nitrogen cycle, converting nitrogen from the air into a form that plants can use. Without nitrogen-fixing bacteria in the soil, plants would not be able to access this essential nutrient. This process is essential not only for agriculture, but also for sustaining ecosystems that depend on plant life for food. A lesser-known fact is that some bacteria are bioluminescent, meaning they produce their own light. These bacteria, such as those from the genus Vibrio, can glow in the dark, creating eerie, luminous effects in the ocean. Some marine organisms have evolved to coexist with these glowing bacteria, using them to attract prey or communicate with potential mates. Bacteria also have a significant role in the human immune system. For instance, Lactobacillus and Bifidobacterium are beneficial bacteria that help maintain the health of the gut, preventing harmful pathogens from taking hold. These good bacteria form part of the microbiome and are often used in probiotics to help balance gut health and support the immune system. Bacterial spores are capable of surviving in a dormant state for incredibly long periods. Bacillus and Clostridium species, for example, can form spores that resist heat, desiccation, and radiation, surviving in harsh environments for centuries. This ability to hibernate makes them incredibly resilient, enabling them to endure conditions that would otherwise be lethal. In some cases, bacteria have been used in biotechnology to create biofuels, biodegradable plastics, and even medicines. Escherichia coli is one of the most well-known bacteria used in genetic engineering, where it is modified to produce substances such as insulin or growth hormones. These advances have revolutionized modern medicine and agriculture. Bacteria can form protective structures called biofilms, which are dense communities of bacteria embedded in a slimy extracellular matrix. Biofilms can form on surfaces such as pipes, medical devices, and teeth, making them difficult to remove and contributing to infections. They are also involved in the natural decomposition of organic matter in water systems. Some bacteria are capable of photosynthesis, just like plants. However, while plants use sunlight, water, and carbon dioxide to produce sugar and oxygen, bacteria like purple sulfur bacteria use hydrogen sulfide instead of water and produce sulfur as a byproduct. This ability allows these bacteria to thrive in environments where oxygen is scarce, such as in deep ocean vents or hot springs. Bacteria have been discovered living in the most extreme environments on Earth. One remarkable example is Thermococcus gametolerans. These bacteria are not only resistant to extreme heat, but also high levels of radiation, making them ideal models for studying life in extraterrestrial environments, where conditions could be similarly harsh. Their survival mechanisms include special enzymes that protect them from heat-induced damage, allowing them to metabolize sulfur and carbon compounds that are abundant in these hostile environments. An astonishing characteristic of some bacteria is their ability to produce a wide array of toxins. Clostridium botulinum, for instance, creates one of the deadliest substances known to science, botulinum toxin. This neurotoxin can cause paralysis by interfering with nerve function, and just a tiny amount is enough to affect an entire population. Despite its danger, botulinum toxin has been harnessed medically in treatments for conditions like chronic migraines and muscle spasms, showcasing how bacteria can be both harmful and surprisingly useful in medical therapies. Bacteria have an amazing ability to swap genetic material. This process, known as horizontal gene transfer, allows bacteria to exchange DNA, which often includes genes for antibiotic resistance. When one bacterium becomes resistant to an antibiotic, it can pass this ability to others, rapidly spreading resistance throughout a population. This phenomenon is one of the main reasons why antibiotic resistance is such a major public health concern. It highlights the need for careful use of antibiotics to slow down this process and preserve their effectiveness in treating bacterial infections. Many bacteria live symbiotically with other organisms, including humans. For example, bifidobacteria and lactobacillus are essential parts of our gut microbiota, helping us digest food and synthesize vitamins like vitamin K. These bacteria break down complex carbohydrates that human enzymes can't digest, converting them into simpler compounds that our bodies can absorb. In return, the bacteria receive a steady supply of nutrients, forming a mutually beneficial relationship. Disruptions to this balance can lead to digestive problems and even affect our immune systems, proving how intimately linked we are to these microorganisms. Some bacteria are capable of photosynthesis, a process they share with plants. However, while plants use sunlight, water, and carbon dioxide, certain bacteria use sulfur or iron to carry out photosynthesis in environments where sunlight is scarce. Purple non-sulfur bacteria are an excellent example, thriving in oxygen-deprived water or soil. They play a crucial role in nutrient cycling, converting carbon dioxide into organic compounds that other organisms can use. Their adaptability allows them to survive in conditions that would be inhospitable to most other photosynthetic organisms, making them valuable players in ecosystems like wetlands and marshes. Bacteria can form intricate and resilient communities known as biofilms. These clusters of bacteria, surrounded by a slimy protective coating, can attach to surfaces such as teeth, medical devices, or pipes. Biofilms provide bacteria with protection from antibiotics, immune responses, and environmental stresses, making infections harder to treat. For example, bacteria responsible for chronic lung infections in cystic fibrosis. Patients often form biofilms in the lungs, complicating treatment. Biofilm formation is a common survival tactic that allows bacteria to thrive in environments where they might otherwise be eradicated, demonstrating their remarkable ability to adapt and persist. The process of nitrogen fixation is critical to life on Earth, and certain bacteria are masters at it. Nitrogen, although abundant in the atmosphere, is not usable by most plants. However, bacteria like Rhizobium can take nitrogen from the air and convert it into a form that plants can absorb. This process is vital for maintaining soil fertility in agricultural systems, reducing the need for chemical fertilizers. The partnership between Rhizobium and leguminous plants, such as beans and peas, is one of nature's most productive examples of mutualism, benefiting both the plants and the bacteria. In the soil, bacteria can influence the health of plants in subtle yet powerful ways. Pseudomonas bacteria, for example, are known to protect plants from diseases by outcompeting harmful pathogens for space and nutrients. These beneficial bacteria secrete compounds that inhibit the growth of harmful bacteria and fungi, acting as natural pesticides. This ability to control plant diseases has made Pseudomonas bacteria a focus of agricultural research, as they could potentially reduce the need for chemical pesticides, leading to more sustainable farming practices. The complexity of bacterial genetics is astounding. Bacteria reproduce through a process called binary fission, where a single cell divides into two identical cells. However, their genetic material is not as straightforward as it might seem. In addition to a single circular chromosome, many bacteria contain plasmids, small circular pieces of DNA that carry additional genes. These plasmids often contain genes that can be transferred between bacteria, such as those for antibiotic resistance or the ability to metabolize unique substances. This genetic diversity makes bacteria incredibly adaptable to changing environments, helping them survive in a variety of habitats. While bacteria are often associated with disease, many are essential for human health. One example is Lactobacillus, a bacterium found in the gut that helps maintain a healthy digestive system. Lactobacillus produces lactic acid, which lowers the pH of the intestines and prevents the overgrowth of harmful pathogens. It also aids in the digestion of lactose, the sugar found in milk, making it crucial for people who are lactose intolerant. The presence of beneficial bacteria like Lactobacillus in our digestive systems is vital for maintaining a balanced gut microbiome and promoting overall health. Bacteria are also incredibly efficient at recycling carbon. In the oceans, marine bacteria play a pivotal role in breaking down dead organic matter, such as plankton, and converting it into carbon dioxide. This process helps regulate the Earth's carbon cycle, which is crucial in controlling the planet's climate. Without bacteria's role in decomposition, much of the organic matter in the ocean would accumulate, leading to an imbalance in the carbon cycle. These tiny organisms are key players in regulating global carbon levels, even though their work often goes unnoticed. In recent years, scientists have discovered that bacteria can see light in ways similar to animals. A group of bacteria known as rhodopsin-containing bacteria use light-sensitive proteins to navigate their environment, a process that was once thought to be unique to animals and plants. These proteins, known as bacteriohodopsins, allow bacteria to absorb light and convert it into energy, much like the way plants use chlorophyll. This discovery has opened new avenues in microbiology and bioengineering, showing that light sensing in bacteria is far more complex and versatile than previously believed. Certain bacteria can harness the power of electricity. Geobacter sulfur-reducens is an electrically conductive bacterium that can transfer electrons to metal surfaces, such as electrodes, in an environment with little oxygen. This capability has inspired research into microbial fuel cells, which use bacteria to generate electricity from organic waste. These bacterial fuel cells could one day provide a sustainable energy source, tapping into the natural metabolic processes of microbes to produce power in a clean and efficient way. In the field of medicine, bacteria are not just pathogens. They are also being used in groundbreaking therapies. One of the most promising areas of research involves bacteriophage therapy, which uses viruses that specifically target and destroy harmful bacteria. Bacteriophages are highly specific to the bacteria they infect, making them a potentially valuable tool in combating antibiotic-resistant infections. Unlike traditional antibiotics, bacteriophages can be tailored to target specific bacteria, offering a more precise and potentially safer alternative in treating bacterial infections. Bacteria play a vital role in the breakdown of organic materials. Decomposing bacteria like Actinobacteria are among the primary agents that recycle dead plants, animals, and organic waste into essential nutrients for the soil. This natural recycling process enriches the soil, ensuring that it remains fertile and capable of supporting plant life. Without these bacteria, organic matter would accumulate, disrupting ecosystems. These microscopic organisms also contribute to the carbon and nitrogen cycles, making their role in environmental sustainability undeniable. In a sense, bacteria are the unseen caretakers of life on Earth, maintaining balance in the natural world. Some bacteria have evolved the ability to survive in highly acidic environments. For instance, Ferroplasma acidarmanus thrives in conditions with a pH as low as 0, an acidity level comparable to battery acid. These extremophiles utilize specialized proteins and enzymes to maintain their cellular integrity despite the harsh conditions. Their resilience has attracted the interest of scientists who study these bacteria for potential applications in industrial processes that require acidic environments, such as mining and waste treatment. The ability to withstand such extreme acidity is a testament to the incredible adaptability of bacteria. Another extraordinary example of bacterial survival comes from the depths of the Antarctic. Here, bacteria have been found thriving in sub-zero temperatures, in conditions that would freeze most life forms solid. Cycrobacter cryohalolentis is one such bacterium. These bacteria have unique adaptations, including proteins that function efficiently in cold temperatures, allowing them to metabolize nutrients and grow even in freezing environments. Their ability to endure such cold conditions has made them valuable subjects in studies on climate change and extremophilic biology. Bacteria's ability to withstand a wide range of temperatures isn't limited to freezing cold. Thermophiles are bacteria that thrive in extreme heat, often found near hot springs and geothermal vents. These bacteria have enzymes that remain stable and active even in high heat, making them invaluable in laboratory applications. One of the most famous enzymes derived from T. aquaticus is Taq polymerase, which is essential in the process of DNA amplification during PCR. polymerase chain reaction, a technique used in genetic research. The bacterial kingdom includes microorganisms capable of performing processes that seem almost magical. Bioluminescent bacteria, such as those in the genus Vibrio, emit light through chemical reactions in their cells. This natural glow, seen in oceanic waters and on some land-based surfaces, serves various purposes in nature. Some species use their light to attract prey, while others use it for communication or to ward off predators. In some species, this ability is even linked to their virulence, with glowing bacteria sometimes indicating the presence of harmful pathogens in marine environments, making them a fascinating study for marine biologists. Interestingly, bacteria can communicate with one another through a process known as quorum sensing. This mechanism allows bacteria to sense the density of their population by releasing and detecting signaling molecules. When the bacterial population reaches a certain threshold, the behavior of individual cells shifts, and they begin to coordinate actions such as forming biofilms or producing toxins. Quorum sensing is critical in the formation of infections, as it enables bacteria to work together and overwhelm the host's immune defenses. This form of microbial groupthink showcases the complexity of bacterial social interactions. Bacteria are central to the process of nitrogen fixation, which is crucial for plant life. Rhizobium species, for example, form symbiotic relationships with the roots of leguminous plants, such as peas and beans. In this partnership, the bacteria convert nitrogen from the air into a form that plants can absorb and use for growth. In return, the plants provide the bacteria with sugars and other nutrients. This process not only helps plants grow, but also reduces the need for artificial fertilizers, making it an essential component of sustainable agriculture. Despite their tiny size, bacteria are incredibly diverse in their abilities. Some bacteria, like those from the genus Bacillus, can produce spores that remain dormant for long periods, sometimes for centuries. These spores are resistant to environmental stresses, such as heat, radiation, and chemicals. When conditions become favorable again, the spores germinate and the bacteria resume their growth cycle. This ability to survive in a dormant state for extended periods enables bacteria to endure harsh conditions, including those found in space, where spores have been shown to survive the vacuum and radiation of outer space. Bacteria's ability to manipulate their environment extends beyond just physical survival. Some bacteria can alter their surroundings chemically. For example, sulfate-reducing bacteria play an important role in the sulfur cycle by reducing sulfate to hydrogen sulfide. This process contributes to the natural sulfur content in the Earth's oceans, marshes, and lakes. In some environments, these bacteria are responsible for the characteristic smell of rotten eggs, produced by the release of hydrogen sulfide gas. Although this gas is toxic, these bacteria are critical in maintaining the balance of sulfur compounds in natural systems. In medical science, bacteria are proving to be both a challenge and a boon. Bacteriophages, viruses that infect bacteria, are being explored as a solution to antibiotic-resistant infections. Unlike antibiotics, which indiscriminately kill bacteria, bacteriophages target specific bacteria, leaving beneficial bacteria untouched. This specificity makes them an ideal candidate for precision treatments. Bacteriophage therapy, first used in the early 20th century but later overshadowed by antibiotics, is seeing a resurgence as scientists explore new ways to combat antibiotic-resistant bacteria in the face of global health crises. A fascinating aspect of bacterial life is their ability to alter their genetic makeup through mutations. While most mutations are random, some are beneficial, giving the bacteria a survival advantage. These beneficial mutations are passed on to future generations, rapidly spreading throughout bacterial populations. The ability to evolve quickly allows bacteria to adapt to changing environments, such as the presence of antibiotics or shifts in temperature or nutrient availability. This rapid evolutionary process is one reason why bacteria can develop resistance to treatments in such a short time, posing challenges for healthcare systems worldwide. Interestingly, bacteria are also capable of eating metals, a process known as biomineralization. Certain species, such as Geobacter, sulfur reducens, can transfer electrons to metals, essentially breathing metal ions instead of oxygen. This ability allows them to survive in environments that are rich in metals like iron and manganese, and it plays a role in the bioremediation of polluted environments. Bacteria can even convert toxic metals like uranium into less harmful forms, making them valuable in the cleanup of contaminated sites. Bacteria have a unique and complex system for protecting themselves from threats. One of the most effective forms of defense is the CRISPR-Cas system, which acts as a bacterial immune system. This mechanism allows bacteria to remember viruses that have previously attacked them, storing snippets of viral DNA in their own genome. If the same virus tries to attack again, the bacteria can use the stored information to recognize and neutralize the threat. This system has been adapted by scientists for gene editing technologies, offering an entirely new approach to genetic modification in research and medicine. In the dark, oxygen-deprived depths of the ocean, bacteria have evolved to use hydrogen sulfide as their primary energy source. These chemotrophic bacteria, such as those in the genus Beggiatoa, can thrive in environments where sunlight cannot reach. By oxidizing hydrogen sulfide, they generate energy in a process called chemolithoautotrophy. This allows them to form thriving colonies around hydrothermal vents and cold seeps, where they act as the foundation of complex ecosystems. These bacteria are crucial in the deep ocean food chain, sustaining not just themselves, but an entire array of life forms that rely on their energy. Bacteria often form intricate, multi-layered communities known as biofilms, which are sticky, protective structures made of bacterial cells and extracellular material. Biofilms can form on various surfaces, including medical devices, water pipes, and even the surfaces of teeth. In fact, the plaque that builds up on your teeth is a form of biofilm. The bacteria within biofilms are harder to kill than individual free-floating bacteria, as the slimy coating protects them from antibiotics and the immune system. This resilience makes biofilms a significant challenge in medicine, often leading to chronic infections that require specialized treatments. One of the most interesting aspects of bacteria is their ability to choose their environment based on external stimuli. E. coli is a well-known example of a bacterium capable of chemotaxis, where it moves toward favorable conditions, such as food sources, and away from harmful ones, like toxic chemicals. This movement is facilitated by flagella, long tail-like structures that propel the bacteria through liquids. By sensing chemical gradients in their surroundings, bacteria can navigate their environment with remarkable precision, ensuring their survival and growth by responding rapidly to changes in their habitat. Bacteria, despite their simplicity, have an impressive ability to adapt to their surroundings. Lactobacillus, for example, is a bacterium commonly found in the human gut. It thrives in the acidic conditions of the stomach and intestines, where it helps break down food and maintain a healthy digestive system. These bacteria are also used in the production of fermented foods like yogurt and sauerkraut, where their ability to produce lactic acid contributes to both the preservation and flavor of the food. Lactobacillus is just one of many bacteria that have specialized to live in close relationship with humans. Some bacteria can harness the power of light to create energy, much like plants do through photosynthesis. But unlike plants, which use chlorophyll, some bacteria use a pigment called bacteriochlorophyll. Bacteria like Chlorobium utilize this pigment to capture light and convert it into chemical energy, which they use to grow and thrive in environments like deep ponds or lakes, where light is scarce. Their unique method of photosynthesis allows them to survive in low-light conditions and is an essential part of the microbial ecosystem in these environments. Certain bacteria, such as Corynebacterium diphtheria, are notorious for producing toxins that cause disease in humans. C. Diphtheria is the causative agent of diphtheria, a serious infection that can lead to difficulty breathing and even death. The bacterium produces a toxin that interferes with protein synthesis in human cells, which is why it can cause such severe damage. However, vaccines have been developed to protect against diphtheria, showing how scientific advancements have turned the harmful capabilities of bacteria into an opportunity to safeguard human health. Some bacteria are capable of producing chemicals that are toxic to other organisms, a process known as antimicrobial production. For instance, Streptomyces, a genus of soil-dwelling bacteria, produces a variety of antibiotics, including streptomycin and tetracycline. These antibiotics can kill or inhibit the growth of other microbes, providing the bacteria with a competitive advantage in their environment. Interestingly, many antibiotics used in medicine today were originally derived from bacterial compounds, underscoring the historical and ongoing relationship between bacteria and the development of medical treatments. Bacteria have evolved strategies for exchanging genetic material that can drastically alter their behavior. Horizontal gene transfer is one such method, where bacteria share DNA through direct contact or by releasing genetic material into their environment. This transfer can confer new abilities, such as antibiotic resistance or the ability to digest a new food source. The process allows bacteria to rapidly adapt to environmental pressures, which is why bacterial populations can evolve so quickly and why the spread of antibiotic resistance is a growing concern in modern healthcare. The unique genetic code of bacteria has allowed them to live in environments that seem completely inhospitable to most other forms of life. For example, bacteria living near deep-sea hydrothermal vents have adapted to survive extreme pressures and temperatures. These extremophiles rely on geothermal heat and the chemical richness of the vents, using sulfur and methane as energy sources. Their enzymes have evolved to function under these harsh conditions, and studying them has helped scientists understand how life could potentially exist on other planets where similar conditions might be found. Some bacteria can act as living factories, producing useful substances for humans. Escherichia coli is one of the most widely studied bacteria and has been genetically engineered to produce a variety of products, from insulin to biofuels. In these applications, bacteria serve as miniature factories, converting simple sugars into complex molecules. This biotechnology revolution has made it possible to produce important chemicals more efficiently and sustainably than traditional methods. The use of bacteria in industrial processes continues to expand, offering a greener alternative to traditional chemical production. The remarkable diversity of bacteria extends beyond their shapes and sizes. They also differ in the environments they can colonize. Mycobacterium tuberculosis, the bacterium responsible for tuberculosis, thrives in the human lungs, where it can evade the immune system for years. By hiding in immune cells called macrophages, it can remain dormant until conditions are right for it to reactivate. This ability to hide and resurface makes M. tuberculosis particularly challenging to treat, as the infection can persist for long periods without causing noticeable symptoms, leading to undiagnosed and untreated cases. Some bacteria are capable of converting carbon dioxide into useful substances through a process called autotrophy. These bacteria, such as those in the genus Rhodobacter, do not rely on sunlight like plants, but instead use other chemical reactions to fix carbon. By utilizing compounds like hydrogen or sulfur, these bacteria can produce organic molecules that are then used by other organisms in the ecosystem. This ability is important for ecosystems in areas where sunlight is not readily available, such as the deep ocean or subterranean environments, ensuring that energy flow and carbon cycling continue. Some bacteria are capable of fixing carbon directly from the atmosphere through a process known as carbon fixation. Acetobacter is one such genus of bacteria that captures carbon dioxide and transforms it into organic compounds through a chemical reaction. This process is essential for maintaining the carbon cycle, as these bacteria help regulate the amount of carbon dioxide in the atmosphere, mitigating the effects of global warming. They play a crucial role in the ecosystems where they reside, contributing to both the survival of plants and the balance of greenhouse gases in the atmosphere. Bacteria's ability to resist environmental threats is often due to their thick cell walls. The cell wall of gram-positive bacteria, for example, is made up of a thick layer of peptidoglycan, which gives them extra protection against physical stress, desiccation, and the immune system. Gram-negative bacteria, on the other hand, have a more complex structure, with an outer membrane that can act as a barrier to antibiotics. These structural differences not only impact how bacteria react to their environments, but also determine how they are treated in medical settings, making bacterial classification vital for diagnosis and treatment. Some species of bacteria can engage in a process known as denitrification, which is crucial in the nitrogen cycle. Bacteria such as Pseudomonas and Paracoccus convert nitrate into nitrogen gas, releasing it back into the atmosphere. This process prevents the overaccumulation of nitrogen compounds in the soil, which could otherwise lead to harmful effects like eutrophication, where excess nutrients cause algae blooms and disrupt aquatic ecosystems. Denitrification by bacteria thus helps maintain a healthy balance in nitrogen levels, benefiting both terrestrial and aquatic environments by recycling nitrogen in a way that other organisms cannot. Many bacteria are opportunistic pathogens, meaning they don't cause disease unless the body's defenses are weakened. Pseudomonas aeruginosa, for example, is a bacterium that is normally harmless in healthy individuals, but can cause severe infections in immunocompromised people, such as those with cystic fibrosis or burn wounds. This bacterium thrives in moist environments like hospitals and can infect various parts of the body, including the lungs, urinary tract, and bloodstream. Its adaptability to different environments and resistance to many antibiotics make it a challenging pathogen to treat in vulnerable patients. Some bacteria are so efficient at living in the human body that they play a central role in our overall health. The gut microbiota, for instance, is composed of trillions of bacteria that help with digestion, immune function, and even mental health. Bacteroides species, which are abundant in the intestines, break down complex carbohydrates into short-chain fatty acids, which are important for maintaining the integrity of the gut lining. These beneficial bacteria also help keep harmful pathogens at bay by outcompeting them for nutrients, making them an essential part of our microbial ecosystem. Bacterial flagella, which are long, whip-like appendages, are essential for the mobility of many bacteria. These flagella rotate like propellers, allowing bacteria to move toward or away from environmental stimuli in a process called taxis. Whether it's moving toward food sources, positive chemotaxis, or away from harmful chemicals, negative chemotaxis, flagella are vital for bacterial survival. Their ability to propel bacteria through liquids efficiently allows them to find optimal living conditions or escape unfavorable environments. The structure and function of bacterial flagella have even inspired designs in robotics and microengineering. In the world of bacteria, size can vary dramatically. While many bacteria are microscopic, some species can grow to sizes visible to the naked eye. Thiomargarita namibiensis, for instance, is the largest known bacterium, measuring up to 0.75 millimeters in diameter, large enough to be seen without a microscope. These bacteria thrive in ocean sediment and can store large amounts of sulfur in their cells, making them crucial players in the sulfur cycle. Despite their enormous size compared to most bacteria, they still exhibit all the hallmarks of bacterial life, including a single-cell structure. Some bacteria can form complex relationships with plants, enhancing their growth and resistance to disease. Azospirillum is one such bacterium that forms a symbiotic relationship with grasses. By fixing nitrogen in the soil, it helps plants like wheat and maize access this essential nutrient, which boosts their growth. In return, the plants provide the bacteria with sugars and other organic compounds. This mutualistic relationship not only improves plant health, but also enhances crop yields, making these bacteria valuable in agriculture, especially in environments with poor soil fertility. In aquatic environments, bacteria are responsible for breaking down organic materials and recycling nutrients back into the ecosystem. Nitrosomonas and Nitrobacter are examples of nitrifying bacteria that convert ammonia into nitrate, a form of nitrogen that plants can use. These bacteria play a crucial role in maintaining the nitrogen balance in aquatic ecosystems. Without them, nitrogen compounds would accumulate, disrupting aquatic life and leading to the formation of harmful algal blooms. Their role in nutrient cycling underscores the importance of bacteria in sustaining life on Earth, from the oceans to the soil. Bacteria are capable of forming spores, which are highly resistant to extreme conditions, such as heat, desiccation, and UV radiation. One of the most famous spore-forming bacteria is Clostridium tetani, which causes tetanus. The spores of C. tetani can remain dormant for years, waiting for the right conditions to germinate. When they find a suitable environment, such as an oxygen-poor wound in a human, they become active and produce toxins that can cause severe muscle spasms. The resilience of these spores makes them particularly dangerous, as they can survive for long periods in the environment before causing harm. Not all bacteria are single-celled. Some are multicellular and can form complex structures. Myxobacteria, for instance, are known for their ability to form large coordinated colonies. These bacteria can move in groups and secrete enzymes that break down organic matter. When conditions become unfavorable, myxobacteria aggregate into fruiting bodies, structures that release spores to disperse and survive until conditions improve. Their ability to form these intricate structures and their highly cooperative behavior make them a fascinating subject for microbiological research. Bacteria's role in human health is not always harmful. In fact, certain species of bacteria are actively used in probiotics, which are supplements containing live microorganisms that promote health. Lactobacillus and Bifidobacterium are two of the most commonly used probiotics, as they support gut health and help maintain a balanced microbiome. These bacteria aid in digestion, support the immune system, and prevent the overgrowth of harmful bacteria in the digestive tract. The widespread use of probiotics is a testament to the valuable, positive impact that bacteria can have on human well-being. In the microscopic world, certain bacteria have the remarkable ability to break down pollutants. Deinococcus radiodurans, often dubbed the world's toughest bacterium, is capable of withstanding extreme radiation, desiccation, and chemical exposure. This resilience makes it a prime candidate for bioremediation, the process of using organisms to clean up environmental contaminants. By absorbing heavy metals or degrading radioactive substances, these bacteria could one day play a critical role in cleaning up polluted sites. Their ability to endure harsh conditions is nothing short of extraordinary, showcasing the untapped potential of bacteria in environmental restoration efforts. There are bacteria that thrive in environments rich in toxic substances, demonstrating incredible adaptability. Ralstonia eutropha, for example, is capable of metabolizing toxic metals like cadmium and nickel. These bacteria possess specialized enzymes that allow them to detoxify and even accumulate metals inside their cells, offering an ecological solution to heavy metal contamination. This capability has led to the exploration of Ralstonia for use in bioremediation, where it could help detoxify contaminated water, soil, or industrial waste sites, turning harmful substances into manageable, less toxic forms. Microorganisms such as Salmonella have evolved complex mechanisms to invade host organisms. These bacteria are capable of infiltrating the intestinal lining, where they can cause infections that disrupt digestion and sometimes lead to serious complications like sepsis. Salmonella utilizes specialized protein complexes to enter host cells and evade the immune system, which allows the infection to spread. These adaptations make the bacterium highly successful in a wide variety of animals, including humans. Studying their mechanisms of infection has provided valuable insight into the ways bacteria can manipulate host cells to gain access to nutrients and proliferate. Among the vast array of bacteria, some possess a unique ability to produce natural antibiotics. Streptomyces species, for example, are soil-dwelling bacteria that produce a diverse range of compounds used in medicine, including the well-known antibiotic streptomycin. These compounds work by inhibiting the growth of other microorganisms, allowing the producing bacteria to dominate in their environment. Streptomyces is not only important for antibiotic production, but also for soil health, as its chemical secretions help suppress the growth of harmful pathogens, maintaining a balanced microbial ecosystem in the soil. In the fight against disease, certain bacterial strains have developed mechanisms to resist antibiotics. Staphylococcus aureus, a notorious pathogen responsible for skin infections, pneumonia, and bloodstream infections, has evolved resistance to many antibiotics, including methicillin. The gene responsible for this resistance, MACE, has spread among different bacterial populations, making infections more difficult to treat. This ability to acquire resistance through horizontal gene transfer makes bacteria like S. aureus formidable adversaries in modern medicine. Ongoing research is focused on finding new treatments to combat these resistant strains, but they remain a significant challenge to healthcare systems worldwide. Bacteria also serve as crucial players in nitrogen cycling in ecosystems. Nitrosomonas and Nitrobacter, two genera of nitrifying bacteria, convert ammonia into nitrites and nitrates, respectively. These nitrogen compounds are essential for plant growth, providing the nitrogen that plants need to form proteins and other vital molecules. Without these bacteria, the nitrogen in the atmosphere would remain unusable by plants, leading to a breakdown in the food chain. These bacteria help keep the nitrogen cycle in balance, ensuring the health of ecosystems ranging from forests to agricultural fields. There are bacterial species that specialize in creating and modifying complex chemical compounds that benefit both themselves and other organisms. Agrobacterium tumefaciens, for instance, is a bacterium known for its ability to transfer genes into plants, causing tumors to form in the process. While this might sound harmful, A. tumefaciens has been harnessed by scientists in genetic engineering. The bacterium's natural ability to insert foreign genes into plant DNA has made it a valuable tool in developing genetically modified crops, such as those resistant to pests or diseases, which could help address food security issues. A fascinating aspect of bacterial life is the phenomenon of antibiotic resistance, which can develop through various means. In addition to gene transfer, bacteria can also develop resistance through genetic mutations that alter the target site of an antibiotic. For example, Escherichia coli can mutate the enzymes it produces so that they no longer bind to commonly used antibiotics, rendering the treatment ineffective. This constant evolution makes antibiotic resistance one of the biggest public health challenges. Understanding these mechanisms is essential for developing new antibiotics and treatments that can outpace bacterial adaptation. In a surprising twist, some bacteria have the ability to produce gases that can influence their environment in subtle yet significant ways. Methanogens, a group of archaea, microorganisms closely related to bacteria, produce methane as a byproduct of their metabolism. Found in environments such as wetlands, the guts of ruminant animals, and landfills, these bacteria play a role in the carbon cycle. While methane is a potent greenhouse gas, it is also an important source of energy for some ecosystems. This process, while contributing to climate change, also underlines the role of bacteria in regulating Earth's atmospheric composition. Bacteria can engage in complex behaviors that involve cooperation and coordination. For instance, Microbacteria are capable of forming intricate multicellular structures when conditions become unfavorable. These bacteria aggregate to form fruiting bodies, structures that contain spores that can survive until environmental conditions improve. This cooperative behavior allows the colony to endure when food is scarce, enabling the bacteria to disperse and colonize new areas. These fruiting bodies are not only a fascinating survival mechanism, but also a reflection of how bacteria can act collectively, showing that microbial life can exhibit more complexity than it initially appears. Bacteria that live in the human gut, such as firmicutes and bacteroidetes, have a profound impact on human health beyond digestion. Recent research suggests that these bacteria influence our metabolism, immune system, and even our brain. The gut microbiome, a diverse community of microbes, plays a role in regulating the gut-brain axis, influencing mood, behavior, and even the susceptibility to diseases like depression and anxiety. By studying the gut microbiota, scientists are uncovering how these microscopic organisms contribute to overall health and may help in the treatment of various mental health conditions. In aquatic environments, bacteria are key players in maintaining water quality. Cyanobacteria, also known as blue-green algae, are capable of photosynthesis, producing oxygen and serving as the base of the food chain in many aquatic ecosystems. While some species of cyanobacteria are beneficial, others can proliferate into harmful algal blooms, especially in nutrient-rich waters. These blooms can deplete oxygen levels, harm aquatic life, and release toxins that affect both wildlife and humans. Studying these bacteria helps scientists understand and manage water quality, offering insights into both ecological health and environmental conservation. Certain bacteria are capable of producing substances that can inhibit the growth of other organisms. Bacillus thuringiensis, a bacterium commonly found in soil, produces toxins that are lethal to insects. These toxins are used in biological pest control, where B. thuringiensis is applied to crops to protect them from harmful insects like caterpillars. The bacterium's ability to selectively target pests without harming humans, animals, or beneficial insects has made it a popular alternative to chemical pesticides, offering a more environmentally friendly solution to managing crop-damaging pests. In extreme environments, bacteria exhibit unparalleled resilience. Halobacterium is a bacterium that thrives in extremely saline environments, such as salt flats and salt mines. These bacteria are classified as halophiles, meaning they require high concentrations of salt to survive. They have specialized proteins and enzymes that help them maintain their cellular functions despite the dehydration caused by the salty conditions. Halobacterium also possesses a unique form of photosynthesis, using light to produce energy in a way that differs from most other organisms, adding yet another layer to the complexity of life in extreme environments. Many species of bacteria have evolved to live in biofilms, structures that protect them from external threats. These slimy colonies can form on any surface that remains wet, such as rocks in streams or medical implants. What makes biofilms so dangerous is their ability to shield bacteria from antibiotics, allowing infections to persist long after treatment. In hospitals, biofilms can form on devices like catheters and prosthetics, leading to chronic infections that are incredibly difficult to treat. The study of biofilm formation and its resistance mechanisms is key to developing more effective medical treatments and preventing persistent infections. One of the most fascinating types of bacteria is the Cyanobacteria, sometimes known as blue-green algae. These bacteria are unique in their ability to perform photosynthesis, producing oxygen as a byproduct. Cyanobacteria were among the first organisms on Earth to release oxygen into the atmosphere, playing a pivotal role in the development of life as we know it. They continue to be integral in aquatic ecosystems, where they serve as primary producers, forming the foundation of the food chain. However, some species can also create harmful algal blooms, which can lead to environmental imbalances. In the world of bacteria, some can communicate with each other in ways that go beyond simple chemical signals. Vibrio fischeri, a bioluminescent bacterium, forms symbiotic relationships with certain marine animals, such as the Hawaiian bobtail squid. The bacteria emit light that helps the squid camouflage itself by matching the brightness of the moonlight above. This glowing is not random. It is the result of the bacteria sensing their population density and synchronizing their bioluminescent activity in a process known as quorum sensing. This form of communication allows the bacteria to work together in a highly organized way. Some bacteria possess the ability to exchange genetic material through a process known as conjugation. This involves one bacterium transferring a small piece of DNA called a plasmid to another bacterium, often conferring new traits such as antibiotic resistance. Escherichia coli, a common bacterium found in the intestines, can participate in conjugation, sharing its genetic material with other bacteria in its environment. This genetic exchange contributes to the rapid evolution of bacterial populations, allowing them to adapt to new environments and resist treatments that would otherwise be effective. Bacteria can be found in some of the most inhospitable places on Earth, and some are capable of surviving without oxygen. Disulfovibrio is a genus of bacteria that lives in environments rich in sulfates, such as deep-sea vents and marshes, where oxygen is scarce. These bacteria use sulfur compounds instead of oxygen in their metabolism, a process known as anaerobic respiration. By metabolizing sulfur, Disulfovibrio plays an essential role in the sulfur cycle, breaking down organic matter and recycling nutrients. This ability to thrive in low oxygen environments makes them crucial in maintaining ecological balance in various ecosystems. Bacteria like Lactobacillus are integral to the fermentation process, where they convert sugars into acids, gases, and alcohols. This process is what allows for the production of foods like yogurt, cheese, and sauerkraut. Lactobacillus and other lactic acid bacteria contribute to the distinct flavors and textures of fermented foods, while also providing health benefits. For example, Lactobacillus can help improve gut health by balancing the microbiome, preventing the overgrowth of harmful bacteria, and aiding in digestion. Fermented foods containing these bacteria are often seen as key components of healthy diets around the world. In environments with little to no light, some bacteria rely on chemosynthesis to produce energy. These bacteria, such as Thiobacillus, use sulfur compounds instead of sunlight to create energy. By oxidizing sulfur, these bacteria are able to create organic molecules that can be used as food for themselves and for other organisms in their environment. This ability to live in the dark and still generate energy has allowed chemosynthetic bacteria to thrive around deep-sea hydrothermal vents, where sunlight cannot penetrate. These ecosystems are entirely dependent on the energy created by these bacteria. Bacterial spores are an ingenious survival strategy used by many species to withstand extreme environmental conditions. Bacillus anthracis, the causative agent of anthrax, produces spores that can lie dormant for years, only to germinate when they encounter favorable conditions. These spores are highly resistant to heat, desiccation, and UV radiation, allowing them to survive in harsh environments. When conditions become conducive to growth, the spores can wake up and begin reproducing, making them an effective means of ensuring the survival of the species even when food and moisture are scarce. In the human body, certain bacteria are crucial for immune system regulation. Lactobacillus and other probiotics help maintain a healthy gut microbiota, which in turn supports immune function. These bacteria train the immune system to differentiate between harmful pathogens and harmless substances, reducing the risk of autoimmune diseases. The gut microbiome has been linked to various aspects of health, from digestion to mental well-being. It is a dynamic, ever-changing community of bacteria that adapts to diet, lifestyle, and other factors, constantly interacting with the immune system to maintain balance and protect the body from infections. The human body hosts trillions of bacteria, many of which are incredibly beneficial. Bifidobacteria, for instance, are integral to maintaining gut health. These bacteria help break down complex carbohydrates that the human digestive system cannot process on its own, producing vital nutrients like vitamins and short-chain fatty acids. They also help prevent harmful pathogens from taking hold in the intestines by competing for resources. The symbiotic relationship between bifidobacteria and the human body is a prime example of how bacteria can play a protective, nurturing role in maintaining overall health and well-being. Another vital group of bacteria in the human body are the lactobacillus species. These bacteria are essential for maintaining the health of the digestive system, particularly in the stomach and intestines. They help balance the gut microbiota, preventing the overgrowth of harmful microbes like Clostridium difficile, which can lead to severe infections. Lactobacillus is also used in the production of fermented foods like yogurt, kefir, and kimchi, where they enhance flavor and promote the growth of healthy microorganisms. Beyond digestion, these bacteria are also believed to play a role in boosting immune function, illustrating their extensive health benefits. While many bacteria contribute positively to human health, others have evolved to resist antibiotics, making them dangerous threats to medicine and public health. Methicillin-resistant Staphylococcus aureus, MRSA, is one of the most infamous examples of antibiotic-resistant bacteria. MRSA has evolved mechanisms that allow it to survive despite the presence of methicillin, a powerful antibiotic once used to treat Staphylococcus aureus infections. MRSA infections are difficult to treat and can lead to serious complications, particularly in individuals with weakened immune systems. The emergence of resistant strains like MRSA underscores the need for ongoing research into new antibiotics and alternative treatments. Antibiotic resistance is a growing concern, as bacteria are becoming increasingly adept at evolving defense mechanisms against drugs designed to kill them. One way this resistance occurs is through genetic mutations that alter the bacterium's cellular machinery, rendering antibiotics ineffective. Escherichia coli, a bacterium commonly found in the intestines, has developed resistance to a wide range of antibiotics, including fluoroquinolones and penicillin. This resistance can spread through horizontal gene transfer, where bacteria exchange genetic material, further complicating efforts to combat infections. The rise of resistant bacteria is one of the most pressing challenges in modern healthcare. The discovery of penicillin revolutionized medicine by providing an effective treatment for bacterial infections. Penicillin, derived from the mold Penicillium notatum, was discovered by Alexander Fleming in 1928. It works by disrupting the bacterial cell wall, causing the bacterium to burst and die. This marked the beginning of the antibiotic era, saving countless lives and transforming the treatment of infections. However, the widespread use of penicillin and other antibiotics has also contributed to the development of antibiotic-resistant bacteria, as some bacteria have evolved mechanisms to resist or neutralize these powerful drugs. The overuse and misuse of antibiotics in both human medicine and agriculture have accelerated the development of resistance. In particular, the routine use of antibiotics in livestock feed has contributed to the emergence of resistant bacteria that can be transmitted to humans through the food supply. This has sparked calls for stricter regulations on antibiotic use in agriculture to prevent further exacerbation of the resistance problem. The situation is compounded by the fact that new antibiotics have not been developed at a pace fast enough to keep up with the evolution of resistant bacteria. Bacteria have evolved a variety of methods to survive antibiotic treatment. One such strategy is the production of enzymes that break down antibiotics, rendering them ineffective. For example, beta-lactamase enzymes are produced by bacteria to neutralize penicillin and similar antibiotics. These enzymes cleave the beta-lactam ring, which is essential for the antibiotic's action, thereby deactivating the drug. The spread of beta-lactamase-producing bacteria has made some infections nearly impossible to treat with older antibiotics, requiring the development of newer, more potent drugs to fight resistant strains. The discovery of penicillin also highlighted the importance of understanding bacterial cell structure. Penicillin targets the cell wall, a feature that is present in most bacteria, but not in human cells. This selective toxicity made penicillin a highly effective treatment for bacterial infections without harming human cells. However, some bacteria, like the Mycobacterium species that cause tuberculosis, have developed thick, waxy cell walls that make it more difficult for antibiotics to penetrate. These bacteria are more challenging to treat and have prompted the development of specialized antibiotics designed to break through their defenses. In some cases, bacteria can share their resistance genes with others through a process known as horizontal gene transfer. This can happen when one bacterium transfers a plasmid, a small piece of DNA, to another bacterium. This plasmid can contain genes that confer resistance to antibiotics, which allows the receiving bacterium to become resistant as well. This process speeds up the spread of resistance and makes it difficult to control infections, as entire populations of bacteria can quickly become resistant to treatment. Horizontal gene transfer is a key factor in the rapid evolution of antibiotic resistance. The overprescription of antibiotics in healthcare settings is another contributor to the rise of resistant bacteria. When antibiotics are prescribed unnecessarily, they kill off sensitive bacteria while allowing resistant strains to survive and proliferate. For example, using antibiotics to treat viral infections like the flu or common cold does not address the root cause of the illness, but instead increases the chances of resistance developing. Health organizations worldwide are urging the responsible use of antibiotics, including only prescribing them when absolutely necessary, to slow the spread of resistance and preserve the efficacy of existing drugs. In response to the rise of resistant bacteria, researchers are exploring alternative treatments that can complement or replace traditional antibiotics. Phage therapy, for instance, uses bacteriophages, viruses that specifically target bacteria, to infect and kill resistant bacterial strains. Phage therapy has been used in certain parts of the world, especially in Eastern Europe, as a potential treatment for infections caused by resistant bacteria. While still in experimental stages in many countries, phage therapy offers a promising solution to the antibiotic resistance crisis by providing a targeted approach that is less likely to lead to resistance. In the deep ocean, bacteria have developed astonishing adaptations to survive in the most extreme conditions. Thermophilic bacteria, such as Pyrolobus fumari, thrive near underwater volcanoes and hydrothermal vents. These bacteria have evolved special enzymes that can function at such high temperatures, making them invaluable for scientific research, including the development of heat-stable industrial enzymes. Their ability to live in boiling water deep beneath the sea highlights the incredible resilience of life in conditions that would destroy most organisms. Some bacteria are capable of consuming oil, which can be especially useful in cleaning up oil spills. Alkanivirax borcumensis is one such bacterium, often found in environments contaminated with hydrocarbons. It has developed specialized enzymes that break down the long chains of hydrocarbons into smaller, more manageable compounds. These bacteria are part of a natural bioremediation process that occurs in the ocean after spills, helping to reduce the environmental impact of petroleum products. Their presence underscores the adaptive potential of bacteria in addressing human-induced environmental disasters. Not all bacteria are harmful to humans. Many play crucial roles in our health and the environment. For instance, Rhodobacter sphaeroides is a bacterium found in soil and aquatic environments that can photosynthesize and fix nitrogen. By converting nitrogen from the air into a form that plants can use, these bacteria play an essential role in the nitrogen cycle. Rhodobacter also has potential applications in renewable energy, as scientists are exploring ways to harness its ability to convert light into energy for sustainable biofuel production. Some bacteria are known for their ability to create sophisticated protective structures called endospores. These spores are incredibly resilient, able to withstand heat, radiation, and desiccation. Clostridium botulinum, which produces botulinum toxin, is another well-known bacterium that forms endospores. These spores can survive for years in harsh environments, only germinating when they find a suitable environment. Their resistance to antibiotics and other forms of treatment makes bacterial spores a serious concern in food safety and clinical settings, as they can remain dormant and then resurface to cause infection. Bacteria have an amazing ability to feel their environment through a process called chemotaxis. By sensing changes in the concentration of certain chemicals, bacteria can move toward or away from these stimuli. Escherichia coli, for example, uses its flagella to swim toward higher concentrations of nutrients, like sugars, and away from harmful substances like toxins. This movement is guided by chemical receptors on the bacteria's surface, making them highly responsive to changes in their surroundings. This ability helps bacteria find optimal environments for growth and survival, which is crucial for their success in diverse habitats. In the soil, bacteria like Nitrobacter play a critical role in converting nitrites into nitrates, which are essential for plant nutrition. This process is part of the broader nitrogen cycle, which maintains the balance of nitrogen in ecosystems. Without these nitrifying bacteria, plants would struggle to access nitrogen, leading to poorer growth and less vibrant ecosystems. The partnership between bacteria and plants highlights the interconnectedness of life and the essential functions that bacteria perform behind the scenes to support terrestrial ecosystems. Bacteria that can utilize light as an energy source are referred to as phototrophic bacteria. These bacteria contain pigments like bacteria chlorophyll, which absorb light and convert it into chemical energy. Rhodospirillum is an example of a phototrophic bacterium that thrives in low-light environments, such as the depths of lakes or ponds. These bacteria not only contribute to the local ecosystem by producing organic matter, but they also play a role in the global carbon cycle, helping to mitigate the effects of climate change by absorbing carbon dioxide during photosynthesis. Some bacteria are known to cause chronic diseases, in part due to their ability to form biofilms. Pseudomonas aeruginosa, a bacterium often found in hospital environments, can form biofilms on medical devices like catheters and ventilators. This ability to form thick, resistant layers of bacterial cells makes infections caused by Pseudomonas particularly difficult to treat. The bacteria's ability to thrive in both moist environments and in human tissues makes it a persistent threat in healthcare, contributing to infections that may be resistant to common antibiotics. Bacteria aren't just small-scale players. They can also have large-scale impacts on the Earth's climate. Methanogens, a group of archaea closely related to bacteria, play a crucial role in producing methane, a potent greenhouse gas in environments like swamps, wetlands, and the guts of ruminant animals. These bacteria produce methane as a metabolic byproduct, and though methane is a significant contributor to climate change, it also serves as a valuable energy source for certain ecosystems. In the context of climate science, understanding the role of methanogenic bacteria is important for efforts to mitigate global warming. Some bacteria can survive by consuming inorganic materials, such as sulfur or iron, through a process called chemolithotrophy. Thiobacillus, for example, is a sulfur-oxidizing bacterium that can use sulfur compounds to generate energy. This process is different from photosynthesis and allows Thiobacillus to thrive in environments where light is unavailable. These bacteria are commonly found in environments like deep sea vents and sulfur springs, where they play a vital role in the sulfur cycle by converting sulfur compounds into forms that other organisms can utilize. Bacteria are constantly evolving, and one of the most fascinating aspects of this evolution is the way they adapt to antibiotic treatments. Streptococcus pneumonia, a bacterium responsible for pneumonia and other respiratory infections, has developed resistance to many classes of antibiotics, including penicillin. This resistance occurs through genetic mutations or the acquisition of resistance genes from other bacteria. The ongoing battle between bacterial adaptation and medical treatment highlights the need for careful antibiotic use, as overuse and misuse can accelerate the development of resistant strains. Not all bacteria are single-celled organisms. Some can form complex multicellular structures. Myxobacteria, for example, are soil-dwelling bacteria that can move together in coordinated groups. When food becomes scarce, they aggregate to form fruiting bodies, releasing spores to spread and find new sources of nutrients. This fascinating behavior, which resembles a form of collective decision-making, highlights the complexity of bacterial life and the remarkable ability of microorganisms to cooperate and adapt in response to environmental pressures. Certain bacteria have evolved the ability to produce bioactive compounds that are useful in medicine. Streptomyces, a genus of soil-dwelling bacteria, is famous for its production of antibiotics like streptomycin, tetracycline, and erythromycin. These antibiotics target specific bacteria, inhibiting their growth or killing them outright. Streptomyces has revolutionized the treatment of bacterial infections, providing a rich source of life-saving drugs. The discovery of antibiotics from these bacteria paved the way for the development of modern medicine, saving countless lives and transforming public health worldwide. One of the most unique characteristics of bacteria is their ability to exchange genetic material through a process called horizontal gene transfer. This exchange allows bacteria to rapidly acquire new traits, such as antibiotic resistance, virulence factors, or metabolic capabilities. In some cases, genes for resistance to specific antibiotics can spread across entire bacterial communities, making infections harder to treat. This phenomenon happens naturally, but human activities like overuse of antibiotics accelerate the spread of resistant genes, creating a pressing public health challenge. Bacteria can form fascinating symbiotic relationships with plants. Rhizobia, a group of nitrogen-fixing bacteria, live in the root nodules of legumes like peas and beans. These bacteria convert atmospheric nitrogen into a form that plants can use, providing them with a crucial nutrient for growth. In return, the plants supply the bacteria with sugars and other organic compounds produced during photosynthesis. This mutualistic relationship is vital for agricultural productivity, reducing the need for synthetic nitrogen fertilizers and improving soil health in sustainable farming practices. Not all bacteria are harmful, and some even play a crucial role in digesting food. The human digestive system is home to a diverse community of bacteria that help break down food, especially complex carbohydrates that the human stomach cannot digest on its own. Bacteroides species, for instance, are responsible for processing dietary fiber and converting it into short-chain fatty acids, which are an important energy source for the body. This microbial community helps extract nutrients from food and supports overall digestive health, highlighting the intricate relationship between humans and the bacteria that inhabit their bodies. In some environments, bacteria can survive by using light for energy without the need for oxygen. These phototrophic bacteria, such as Chromatium species, thrive in environments like lakes, ponds, and marshes where sunlight penetrates, but oxygen is scarce. Using pigments like bacteria chlorophyll, these bacteria capture light energy and convert it into chemical energy. This process allows them to live in anaerobic conditions, making them some of the earliest life forms to harness solar power long before plants and algae evolved to perform photosynthesis. Certain bacteria have evolved ways to survive in environments with extreme acidity or alkalinity. Acidithiobacillus ferrooxidans, for example, thrives in highly acidic environments, such as sulfuric hot springs and acid mine drainage. It can oxidize iron to obtain energy, a process that creates sulfuric acid as a byproduct. These bacteria are not only fascinating for their resilience in extreme conditions, but they are also used in bioleaching, an industrial process where bacteria help extract valuable metals, such as gold and copper, from ore. Bacteria play an indispensable role in the process of decomposition. Actinobacteria, including the genus Streptomyces, are key players in breaking down complex organic materials in the soil, recycling nutrients back into the ecosystem. They decompose plant matter, dead animals, and other organic substances, transforming them into simpler compounds like carbon dioxide and minerals that plants can use for growth. Without these decomposers, the Earth would quickly become overwhelmed with waste, and nutrient cycling would break down, leading to a collapse of ecosystems. Some bacteria are incredibly resourceful in their ability to produce energy from chemicals that most other organisms would consider toxic. Geobacter sulfur-reducens is a bacterium that can use toxic heavy metals, such as uranium, as an energy source. It transfers electrons to metals, which allows it to generate energy. This remarkable ability has garnered interest in the field of environmental cleanup, as Geobacter may one day be used to help clean up polluted sites by converting toxic metals into less harmful forms. Their unique method of metabolism opens new possibilities for bioremediation techniques. In the context of extreme survival, Deinococcus radiodurans stands out for its ability to withstand radiation levels that would kill most living organisms. This bacterium is known for its remarkable DNA repair mechanisms, which allow it to survive exposure to ionizing radiation, desiccation, and even certain toxic chemicals. Its ability to repair its DNA efficiently after being damaged by radiation has earned it the nickname Conan the Bacterium. Researchers are exploring the potential of Deinococcus for use in environmental cleanups and space exploration, as its durability may be invaluable in hostile environments. Some bacteria can eat metals, a phenomenon known as biomining. Thiobacillus ferrooxidans, for example, can oxidize iron and sulfur compounds to derive energy, a process that is central to bioleaching, an industrial method used to extract valuable metals from ores. This ability allows bacteria to consume certain minerals, helping to break down rocks and release metals such as copper and gold. This process has revolutionized mining, offering a more environmentally friendly alternative to traditional methods, which can cause significant environmental damage. In the world of bacteria, cyanobacteria are often referred to as the progenitors of life on Earth. These organisms were among the first to perform oxygenic photosynthesis, producing oxygen as a byproduct. This led to the Great Oxygenation Event, a critical moment in Earth's history when oxygen levels in the atmosphere began to rise, paving the way for the evolution of complex life. Cyanobacteria are essential essential players in aquatic ecosystems, helping to oxygenate water and serving as the foundation of food webs in oceans, lakes, and rivers around the globe. Bacterial species like Pseudomonas aeruginosa are known for their ability to survive in hospital settings, often infecting individuals with weakened immune systems. These bacteria are highly resistant to many common antibiotics and can form biofilms on medical devices, making them especially difficult to treat. P. aeruginosa. Infections can range from minor skin rashes to more severe respiratory infections or even sepsis. The bacteria's ability to adapt to diverse environments, including human tissues and medical equipment, makes them a challenging pathogen for healthcare professionals to manage. Some bacteria have evolved to breathe substances other than oxygen. Denitrifying bacteria, such as Pseudomonas denitrificans, use nitrate as an electron acceptor in their metabolism, allowing them to survive in oxygen-poor environments like soil and water. These bacteria are vital in the nitrogen cycle, as they convert nitrates back into nitrogen gas, releasing it into the atmosphere. This process helps maintain the balance of nitrogen in ecosystems and ensures that nitrogen compounds do not accumulate to toxic levels in the environment. Bacteria can communicate with each other through a process known as quorum sensing. This allows them to coordinate group behaviors, such as the formation of biofilms or the production of virulence factors, based on their population density. For instance, Vibrio fischeri, a bioluminescent bacterium, uses quorum sensing to regulate the intensity of light it produces. This light helps the Hawaiian bobtail squid camouflage itself, providing a mutual benefit. The ability of bacteria to communicate and collaborate in this way is a striking example of microbial intelligence. Some bacteria can breathe metals instead of oxygen, a process known as metal respiration. Geobacter sulfur-reducens is one such bacterium that can use metals like iron and uranium to extract energy. By transferring electrons to these metals, it generates an electrochemical gradient that powers its metabolism. This ability makes Geobacter incredibly useful in environmental cleanup, where it can help to reduce toxic metals such as uranium in contaminated sites. Their natural talent for breaking down harmful substances may one day be harnessed for sustainable bioremediation, turning hazardous waste into manageable forms. Bacteria can survive under some of the harshest conditions on Earth, and one of the most extreme environments they inhabit is the deep-sea hydrothermal vent. In these vents, where the pressure is immense and water is saturated with toxic chemicals, yet bacteria like Thermococcus and Pyrolobus fumara not only survive, but thrive in these hot, sulfur-rich waters. These bacteria have specialized enzymes that remain functional in extreme heat, making them valuable for scientific research into industrial processes and biotechnology. Bacteria can adapt to their environment through a process called genetic variation, where changes in their genetic material lead to new traits. This variation allows bacteria to survive in diverse habitats, from the deepest ocean trenches to the hottest springs. Escherichia coli, a bacterium commonly found in the human gut, can evolve quickly to tolerate harsh conditions. For example, in response to a lack of nutrients, E. coli can alter its metabolic pathways to make use of alternative food sources, demonstrating the remarkable flexibility of bacteria in the face of adversity. One of the most fascinating ways bacteria can adapt is through the formation of biofilms. These slimy, protective layers of bacteria attach to surfaces and allow the bacteria to grow and thrive in environments where they would otherwise struggle to survive. Pseudomonas aeruginosa, for example, is notorious for forming biofilms on medical devices such as catheters and ventilators. The biofilm serves as a shield, protecting the bacteria from antibiotics and immune system defenses, which makes infections particularly difficult to treat. The study of biofilm formation is essential to developing more effective therapies. While bacteria are known for their resilience, they are not invincible. In fact, many bacteria can be killed by exposure to UV light. Some bacteria have evolved protective mechanisms to shield themselves from UV radiation, but others, like Deinococcus radiodurans, have become exceptionally resistant. Deinococcus is often referred to as the world's toughest bacterium due to its ability to withstand extreme levels of UV radiation, desiccation, and even ionizing radiation. Researchers are studying Deinococcus for its potential use in space exploration, where its radiation resistance could be invaluable in protecting equipment and even human missions from cosmic rays. Some bacteria can smell their environment using specialized receptors. Bordetella bronchiseptica, a bacterium responsible for respiratory infections in animals, has an advanced system for detecting chemical signals in the air. This ability to sense volatile compounds allows bacteria to find favorable environments for growth. For instance, B. bronchiseptica can detect the presence of animal hosts by smelling specific chemicals in the host's breath or skin. This chemosensory capability is a key survival tactic, guiding bacteria to areas where they can thrive and propagate. Bacteria often have unique survival strategies to compete for resources in crowded environments. Streptomyces, a genus of soil-dwelling bacteria, produces a wide array of antimicrobial compounds, such as streptomycin and tetracycline, to fend off competing microorganisms. These naturally occurring antibiotics help Streptomyces secure territory in nutrient-rich environments by inhibiting the growth of nearby bacteria. Interestingly, many of the antibiotics used in modern medicine were first discovered in Streptomyces, making these bacteria invaluable to the development of pharmaceuticals and treatments for bacterial infections. In the human body, bacteria play a significant role in immune system regulation. The gut microbiome, a complex community of bacteria in the intestines, influences how the body reacts to infections and inflammation. Certain strains of Lactobacillus and Bifidobacterium can modulate the immune system, enhancing the body's ability to fight off infections and reducing the risk of chronic diseases. This connection between bacteria and immunity highlights the importance of maintaining a balanced gut microbiome for overall health, as disruptions to this community can lead to autoimmune disorders and other health complications. Some bacteria are capable of producing natural antibiotics that can target specific pathogens. Bacillus subtilis, a soil bacterium, is known for producing a wide range of antimicrobial compounds. One of these, subtilosin, has been shown to inhibit the growth of pathogenic bacteria, making B. subtilis a natural form of antibiotic defense. Interestingly, Bacillus subtilis is also a common bacterium in the human gut, where it helps maintain microbial balance by keeping harmful bacteria in check. The study of these naturally occurring antibiotics is essential for discovering new treatments as antibiotic resistance becomes more widespread. Bacteria have a remarkable ability to process waste materials, especially organic matter. Pseudomonas putida, a bacterium commonly found in soil and water, is capable of breaking down toxic substances like benzene, toluene, and xylene, compounds found in pollutants like gasoline and industrial waste. This ability to degrade harmful chemicals makes P. putida an important player in bioremediation efforts, where bacteria are used to clean up contaminated sites. The bacteria use these toxic compounds as a food source, transforming them into harmless byproducts like carbon dioxide and water, helping restore environmental balance. Many bacteria have evolved mechanisms to resist environmental stresses, including drought, extreme temperatures, and even heavy metals. Cupriovidus metallidurans, a bacterium found in contaminated environments, can survive exposure to toxic metals like gold, mercury, and copper. This bacterium uses specialized enzymes to detoxify the metals, turning them into less harmful compounds. C. Metallidurans is a prime example of how bacteria have evolved ingenious strategies for survival, and it has been studied for its potential use in environmental cleanups where metal contamination is a problem. Some bacteria are masters of survival, capable of living in the vacuum of space. Deinococcus radiodurans, often dubbed the toughest bacterium on Earth, has survived exposure to the harsh conditions of space. In 2007, scientists sent D. radiodurans aboard a spacecraft, where it endured intense radiation, dehydration, and the vacuum of space. Upon returning to Earth, many of the bacteria remained alive and capable of reproducing. This incredible resilience is due to its robust DNA repair mechanisms, making it a candidate for future space exploration, where radiation exposure is a major concern. Bacteria like Vibrio cholerae, the causative agent of cholera, can spread through contaminated water and wreak havoc on communities. Cholera is primarily spread through the consumption of water or food contaminated with the feces of infected individuals. Once ingested, the bacteria colonize the small intestine, producing toxins that cause severe dehydration, diarrhea, and potentially death if untreated. Despite the devastating effects, advancements in water sanitation and antibiotic treatment have made cholera largely preventable. Still, outbreaks continue to occur in areas with inadequate sanitation infrastructure, highlighting the importance of clean water access. Not all bacteria cause harm. Many are vital for nitrogen fixation in the soil. Azotobacter, a free-living bacterium, is an essential player in the nitrogen cycle, converting atmospheric nitrogen into a form that plants can use. This natural process reduces the need for synthetic fertilizers, which are often harmful to the environment. By enriching the soil with usable nitrogen, Azotobacter plays a key role in supporting agricultural productivity and promoting healthy ecosystems. These nitrogen-fixing bacteria also help maintain the balance of nitrogen in the environment, preventing the buildup of excess nitrogen that could disrupt ecosystems. The human microbiome is home to a staggering variety of bacteria, each playing a specialized role in maintaining health. Faecalibacterium prausnitzii is a bacterium found in the intestines that has anti-inflammatory properties. It is believed to help regulate the immune system and maintain the integrity of the intestinal lining. Research has shown that F. prausnitzii is beneficial for gut health and may help prevent conditions like inflammatory bowel disease, IBD. The balance of bacteria in the gut is crucial for overall well-being, demonstrating the deep connection between microbes and human health. Bacteria also play an essential role in the global carbon cycle. Clostridium species, which can break down plant material, are involved in converting organic carbon into methane, a potent greenhouse gas. While this may seem harmful, methane produced by bacteria is an important natural part of the carbon cycle. In fact, bacteria like Methanobacterium are responsible for producing the methane found in wetlands, marshes, and even the guts of ruminant animals like cows. This methane is then released into the atmosphere, where it contributes to global warming. But it also plays a role in the recycling of carbon in nature. Bacteria have developed some of the most effective tools for breaking down organic matter. turning dead plants and animals into nutrients for new life. Bacillus species are a group of bacteria that play a crucial role in decomposing organic material. These bacteria are found in soil and are responsible for breaking down proteins, fats, and other complex molecules. Their enzymatic breakdown of organic matter returns vital nutrients like nitrogen, phosphorus, and carbon to the soil, enriching it and supporting the growth of new plants. This process is fundamental to nutrient cycling and soil health. Bacteria can be programmed to perform specific tasks through genetic engineering, opening up a wide range of possibilities in biotechnology. By altering the genetic material of bacteria, scientists can create bacteria that produce valuable substances, such as insulin, vaccines, or even biodegradable plastics. For instance, Escherichia coli has been genetically modified to produce human insulin, a breakthrough in the treatment of diabetes. The ability to engineer bacteria to perform specialized tasks has vast implications for medicine, environmental cleanup, and industry. In extreme environments, some bacteria have evolved to use sulfur compounds as their energy source. Thiobacillus species, for example, thrive in environments such as sulfur springs and hydrothermal vents, where oxygen is scarce. These bacteria oxidize sulfur compounds to extract energy, a process known as sulfur oxidation. This allows them to survive in places where few other organisms can. These sulfur-oxidizing bacteria play an essential role in the sulfur cycle, helping to break down sulfur-rich compounds and recycle them within the ecosystem. The ability of bacteria to form spores is one of nature's most remarkable survival strategies. Clostridium tetani, the bacterium responsible for tetanus, forms spores that are capable of surviving harsh environmental conditions for years. These spores are resistant to heat, drying, and chemicals, allowing the bacterium to persist in the soil, where it can later infect humans if they suffer a puncture wound. The process of spore formation is a key evolutionary adaptation that allows bacteria to withstand periods of environmental stress and thrive when conditions improve. Certain bacteria have evolved to eat plastic, a feat that may one day help alleviate the global plastic pollution crisis. Ideonella sakaiensis is a bacterium discovered in Japan that can break down polyethylene terephthalate, PET, a plastic commonly used in bottles. This bacterium uses enzymes to degrade PET into its simpler building blocks, which it then consumes. Although this process is slow, the discovery of such plastic-degrading bacteria has prompted significant research into the possibility of using these organisms for bioremediation to tackle plastic waste. Bacteria that live in extreme temperatures, such as those found in volcanic hot springs, are called thermophiles. Thermophiles have specialized proteins and enzymes that can withstand high temperatures. One such bacterium, Thermus aquaticus, produces an enzyme called Taq polymerase, which is essential in the process of polymerase chain reaction, PCR. PCR is a laboratory technique used to amplify DNA, and the enzyme's heat tolerance makes it ideal for the high temperatures required in this process. The discovery of Thermus aquaticus has been pivotal in advancing genetic research. In the ocean, some bacteria play a crucial role in producing oxygen through photosynthesis. Prochlorococcus, one of the smallest and most abundant photosynthetic organisms on Earth, is found in the sunlit regions of the ocean. This bacterium accounts for a significant portion of the Earth's oxygen production, making it a key player in the global oxygen cycle. Despite being invisible to the naked eye, Prochlorococcus has a profound impact on life on Earth, contributing to the oxygen we breathe and supporting marine food chains. Some bacteria have evolved to withstand the toxic environments created by heavy metals. Cupriavidus metallidurans, a bacterium often found in contaminated soils, can tolerate and even thrive in the presence of toxic metals such as gold, mercury, and copper. It does this by transforming these metals into less harmful forms, or by storing them inside its cells. This ability has made C. metallidurans a subject of interest for bioremediation, as it could one day be used to help clean up environments polluted with heavy metals, turning hazardous substances into something more manageable. Certain bacteria can communicate with each other through chemical signals, a process called quorum sensing. By releasing signaling molecules into their environment, bacteria are able to coordinate their behavior, including the formation of biofilms or the production of toxins. For example, Vibrio fischeri, a bioluminescent bacterium, uses quorum sensing to regulate the intensity of light it produces, which benefits both the bacterium and its host, the Hawaiian bobtail squid. This kind of communication is crucial for the survival and cooperation of bacterial communities in nature. Some bacteria have developed a fascinating strategy for survival known as cyst formation. In this process, bacteria like Azotobacter form protective, dormant cysts that allow them to endure extreme environmental conditions such as drought or high salinity. These cysts are highly resistant to heat, chemicals, and radiation. Once the conditions improve, the cysts can germinate, and the bacteria resume their normal activity. This ability to hibernate during unfavorable times is a key survival tactic for many species of bacteria in harsh environments. The relationship between bacteria and fungi is often one of competition, but it can also be mutually beneficial. Myxobacteria, for example, are known for their ability to cooperate with fungi in breaking down organic matter. These bacteria produce enzymes that help decompose complex substances such as cellulose and lignin, which are often too tough for other organisms to process. In return, the fungi provide nutrients that the bacteria can use to fuel their metabolic processes. This cooperative relationship is crucial in ecosystems where decomposing plant material is a primary source of energy. The discovery of bacteria that can survive on rocks floating in space has opened new doors for astrobiology. Bacillus species, among others, have been shown to survive cosmic radiation, extreme temperature fluctuations, and the vacuum of space when encased in meteorite-like materials. This resilience suggests that bacteria may be capable of surviving interplanetary travel, potentially leading to the spread of life beyond Earth. If life on Earth is found to have extraterrestrial origins, it may well have been carried across space by bacterial spores. Lactobacillus is a genus of bacteria that plays an essential role in fermentation processes, such as in the production of yogurt, cheese, and sauerkraut. These bacteria convert sugars into lactic acid, a process that not only preserves the food, but also gives it its characteristic tangy flavor. In addition to their culinary contributions, Lactobacillus species are also beneficial to human health, promoting the balance of gut microbiota and supporting the digestive system. Their presence in fermented foods is a long-standing tradition that connects the worlds of food production and microbiology. One remarkable ability of bacteria is their capacity to adapt to various energy sources. Ralstonia eutropha, for instance, can use a range of organic and inorganic compounds for energy, including hydrogen gas, carbon dioxide, and fatty acids. This metabolic flexibility allows our eutropha to survive in environments where other organisms might not, making it a prime candidate for research in bioenergy production. By harnessing its ability to metabolize hydrogen, scientists hope to develop sustainable energy sources, opening the door to greener technologies for the future. Some bacteria can produce enzymes that break down plastics, which could play a role in combating the growing plastic waste problem. Ideonella sakaiensis is a bacterium discovered in Japan that feeds on polyethylene terephthalate, PET, a type of plastic commonly found in bottles. This bacterium uses specialized enzymes to degrade PET into its basic components, which it then consumes. Though the process is slow, the discovery of plastic-degrading bacteria is an important step toward finding natural solutions for managing plastic pollution. The ability of bacteria to form spores is a crucial survival mechanism that allows them to persist in extreme environments. Clostridium difficile, the bacterium responsible for severe gut infections, forms spores that can withstand heat, drying, and exposure to disinfectants. These spores can remain dormant for long periods, only becoming active when conditions are right, such as when antibiotics disrupt the gut's normal bacterial balance. The spore-forming capability of C. difficile makes it a particularly persistent pathogen, often requiring targeted treatments to eliminate. Bacteria are often referred to as the planet's first life forms, and for good reason. Fossil evidence suggests that bacteria were the first organisms to appear on Earth, existing at least 3.5 billion years ago. The earliest bacteria were simple, single-celled organisms that relied on basic processes like fermentation and photosynthesis to produce energy. Over time, bacteria evolved into a vast array of forms, from the pathogens that cause diseases to the beneficial microorganisms that sustain ecosystems and human life. Their adaptability and resilience have allowed them to thrive in almost every corner of the Earth. As our journey through the microscopic world of bacteria comes to a close, allow yourself to relax even deeper. Let the calmness of this invisible realm wash over you as the tiny organisms we explored continue their quiet dance in the background of life. Feel the peace that comes from understanding their role in shaping our world, knowing that everything, no matter how small, is part of a greater whole. Let your body soften, your thoughts drift away, and your breath becomes slow and steady. If you enjoyed this relaxing moment, please consider leaving a like, subscribing, and turn on all notifications so you don't miss any of our future videos. But for now, let your body relax, release any tension, and allow your mind to drift. Good night and sleep well.